Welcome back for another class on ribozymes, and we left here in the last class, that is structure of hammerhead ribozyme. As you can see from this picture itself, that it resembles the head of hammerhead shark, hence the name hammerhead ribozyme is given. So what I would say is that these kind of molecules, which are named after different shapes, are quite handy for the functioning of various biological functioning of the cell, and we will see them more in detail. So, some features of the hammerhead ribozyme. It's an RNA molecule that catalyzes reversible cleavage and joining reactions at specific site within an RNA molecule. So, the major function is reversible cleavage and joining. That means it causes the cleavage of nucleic acid in a reversible manner, and it's a model used for targeted RNA cleavage experiments and with potential therapeutic application. That is why you will find lots of research work going on with hammerhead ribozyme, because you can literally engineer them and you can use them for targeting some viral RNA, etc., which is infected into a host. And as I already told you, named after the shape, the resemblance to the head of a hammerhead shark, and at molecular level it resembles so. And hammerhead ribozymes RNAs are originally discovered in two types of plant virus-like RNA, that is satellite RNA and viroids. And they also have been found to be widely dispersed within many forms of life forms such as insects, plants, and mammals. So it is not restricted to just plants. They are seen in other animals also. So you can see more in detail about the hammerhead ribozyme structure and its catalysis and gene regulation in this article. Those who are interested can read more in detail about this hammerhead ribozyme. And hammerhead and hairpin ribozymes can be found in several satellite RNAs associated with plant viruses. Majority of the plant viruses are RNA viruses. One example is tobacco ring spot virus, and they do have the hammerhead ribozyme. And it is also has been known to be embedded in the UTRs. UTR stands for untranslated region. UTRs of several mammalian RNAs. And now moving on to another ribozyme, that is hepatitis delta virus. In short form, it is called HDV. It is a satellite RNA of the hepatitis B virus or HBV. Hepatitis B virus is a very important and quite problematic viral infection that infects the liver, and it is as important as HIV in terms of its spreadability through blood. So it is more infective than even HIV. Like you can spread, it's quite contagious, but of course it passes through body fluid, especially blood. So HDV is a human pathogen, co-infection of HDV with HBV. Remember, hepatitis delta virus along with hepatitis B virus is more severe than the infection of HBV alone. Or in other words, if a person has got HDV alone, it is less likely to be detrimental or crucial for the life of an organism. Whereas if that person encounters an HBV, hepatitis B virus infection, then things can go really wrong. HDV is the only catalytic RNA known to be required for the viability of a human pathogen. What is that pathogen? HBV. And is the fastest known naturally occurring self-cleaving RNA, and its rate is around 52 reactions per minute. It's one of the fastest replication rate. And nested double pseudo knockout, that is the structure what it follows for the breaking of the nucleic acid, in which the reactive site is remaining buried. So this nested double pseudo knot is the structural feature what we reflect, as you can see here in the right hand side in this picture, and this structure allows this faster rate of reaction, roughly around 100-fold faster than hammerhead ribozymes. Hammerhead ribozymes also reasonably faster, but HDV ribozyme is much faster. The structural feature called nested double pseudo knot is quite capable of imparting this fast reaction. For moving on to more details about HDV, the hepatitis delta virus is a non-coding RNA found in the hepatitis delta virus that is necessary for the virus replication. And it is thought to be the only catalytic RNA known to be required for the viability of human pathogen. So this catalytic RNA part is very important for the viability of the human pathogen such as HBV. The ribosome acts to the process the RNA transcript to unit length by self-cleavage reaction. That means, as we saw in the previous class, it is almost like a circle, like a bangle, and a copy has to be formed from the existing single-stranded RNA template. And now it continues in a rolling circle fashion. And this ribosome is found to be active in vivo in the absence of any protein factors and is the fastest known naturally occurring self-cleaving RNA. So this is how it occurs. This red colored circle is the entire ribosome in the circular form, and now the blue arrow is the copy. That is, the red one is the template and blue is the copy, and it completes one circle, and then it starts replacing this already formed old newly formed strand, and it comes out of the circle, and this is called a rolling circle replication. As it continues, it needs to have a feature to cut at uniform distance, and this will now circle together and form a independent bangle-like structure as you see here. So this cutting at specific loci, specific distance happens because of the structural features embedded within. Just like group 1 and group 2 introns, we saw the structural feature is lying within the sequence itself. No one comes from outside, no protein comes from outside. So that is what you should keep in mind, that ribozymes act on their own. Now moving on to riboswitches. Riboswitches are elements of bacterial mRNA that control gene expression via binding of small molecules such as coenzyme, amino acid, nucleobases. So they are sequences present in the bacterial mRNA. They are embedded inside, but they can control the expressivity of this mRNA. One example is GLMS ribosome, that is located in the 5' untranslated region of an mRNA. That mRNA codes for glucosamine 6-phosphate, abbreviated as GLCN6P synthetase. So glucosamine 6-phosphate synthetase is the enzyme whose 5' untranslated region has this GLMS ribosome. In the presence of GLCN6P product, that is the product of this mRNA, it cleaves its own mRNA, which downregulates the production of the synthetase. That means if the product is present, then it will recognize this 5' untranslated region ribosome sequence. When the product is there, that ribosome will recognize the product. Once it is recognized, it will cause the degradation of the mRNA so that no more new product is formed. So riboswitches may have functioned as the metabolite sensors in primitive organisms. That means demand and supply. It's like if you are eating food and if your plate has got enough food, no point in keep on delivering food, because then it will start spilling out. You won't be able to eat. So if those plates are empty, I should be able to, or anyone else should be able to serve food. So this is the logic. If the product is there, mRNA should not keep producing more and more product. So the mRNA should be degraded. And when the product is less, then the mRNA should not be degraded. Remember, mRNA is constantly being produced from the organism in those cases where the mRNA is regulated. So let us look into the mechanisms of riboswitch catalyzed reaction. First one is a conformational regulation. So the metabolite binding induces, that means in the previous case, the 5' untranslated region has the ribosome sequence and the product goes and binds onto it. And so the metabolite binding induces a conformational change in the RNA that affects the transcription termination or translation initiation. So, in some case, it is the conformational change induced in the mRNA because of the metabolite binding onto it. Second case is a chemical. In this case, GLMS. What happens? GLC N6P amine might serve as an acid to activate the leaving group after the cleavage. So, this change happens because once the degradation of the RNA happens, the leaving out product should be stabilized, without which the bacterial system can suffer. So that is a chemical means of stabilizing the leaving group. As you can see here, the bond which is cleaved is given in orange in color. So once the product, this is the GLCN6P, which comes and binds onto the target where the ribosome sequence is there, and it can cause the cleavage, and this product stabilizes the leaving group so that there will not be any unwanted effect, and there is no repairing of this RNA and returning back of this mRNA should not happen. So that is the idea. So remember, riboswitch can catalyze in two different ways. One is conformational and another is purely chemical means. So ribosome is another example of a ribozyme which we have seen in the earlier classes also, but in this context we have to quickly touch upon the ribosome also. So the function of the ribosome is peptidyl transferase. That means it is creating a peptide bond, and that is done by the ribozyme action of the ribosomal RNA. So what you can see here, during translation there has to have, like I told you, there are two sites. One is an A site, acceptor site, P site, peptidyl site or peptidyl transferase site. So two sites are there in the ribosome, and it has got two units. One is a large subunit and a small subunit, and the middle what you are seeing is the mRNA. What you see, this green color small structure is a tRNA, and this yellow ball is the amino acid. So in the acceptor site, this tRNA amino acid complex comes in through the acceptor site, and we call it as amino acid tRNA, which is an amino acid containing tRNA, which comes inside, and this always the translation is started by AUG. AUG is the first base which brings in the methionine, which is already here through the acceptor site, that has already come. Now the second one, UCA. So a serine is coming in UCA, which enters in here. Then what happens? It moves inside and binds onto the codon, and the tRNA has got a sequence called anticodon, and it binds in here. And now this peptide synthesis has to happen between the methionine and the serine. MET and SCR. Methionine is an amino acid, serine is another amino acid. It has to form the peptide bond, which is done by the ribozyme action. Then what happens? There will be translocation. That means this methionine is now jumping onto the serine by peptide bond formation, and the methionine containing transfer RNA is now expelled out, and now it is present in the P site. Now it is ready for the another amino acid containing tRNA to enter into the A site. So what happened? Peptide bond is formed, and then also it caused a jumping. That means the serine containing tRNA is no more sitting in the A site, but it jumped down to the P site and making the A site empty, so that newer tRNA containing amino acid can come inside, and the peptide synthesis can continue, and eventually you have a long protein. So this is the structure of the ribosome. This also we saw in the earlier class, that mainly it has got a site and it has got a P site, which is revealed from the structural crystallography studies. So the peptidyl transferase activity can be enhanced by a protein called L27. L27 stands for a ribosomal protein which do not participate in the peptide bond formation, but rather it is necessary for enhancing the activity. However, even in the absence of this protein, which protein? L27 protein, reduced activity can still be observed. Although this protein facilitates peptide bond formation, it is not essential for the peptidyl transferase activity of the ribosome, because which indeed is carried out by an RNA molecule. So how does the RNA catalyze peptide bond formation? Base pairing between the CCA. CCA, remember, it is a tip of the transfer RNA which is formed by the post-transcriptional modification. So the base pairing between the CCA end of the tRNA in the P and A site. Remember, P is the peptidyl site and A is the acceptor site. And the 23S ribosomal RNA, which is part of a ribosome itself, help in the position of the alpha amino group of the amino acid tRNA to attack the carbonyl group of the growing polypeptide. So this is the chemistry that happens during the peptide bond formation. Now, the proton transfer from the amino group of the amino acid tRNA via the 2' OH. Remember, 2' carbon's hydroxyl group is highly reactive. It is helpful in the splicing and various other biochemical reactions. And this 2' OH of the adenosine, that adenosine is belonging to the CCA. From the terminal CCA, C and C stands for 2 cytosine residue, and then you have got a adenine residue. And this adenine residue's 2' OH is participating, and that of this tRNA is helpful in the P site, that is present attached onto the P site. And what happens? This 2' OH to the O3' that is of the accompanied by the peptidyl group of, or in other words, peptidyl group is written as CO-R. R is basically a common group. CO is the peptide bond, peptidyl or a carbonyl bond, we call it as that, and transfer to the amino acid tRNA. So this proton transfer from the amino group of the amino acid tRNA via the 2' OH of the adenosine to its oxygen at the 3' of the COR group is what is going to contribute to this peptide bond formation. Now, coming back to RNA world hypothesis. RNA initially served both as a genetic material and catalyst. Later, the catalytic function of many RNA molecules were taken over by proteins as a convenience, because proteins can adopt much diverse structure and it also have got 20 amino acids to play with, unlike the ribosome. So hence protein became quite handy. So RNA world was there, that time itself the proteins were handy, by certain RNA molecules were capable of doing this job. So various cationic clays such as monomorillonate clay can promote the polymerization of RNA-like monomers into RNA chains. So this is what would have happened in the RNA world. So RNA is the primary substance of life, and DNA and proteins are later refinements for better performance of this RNA's function or RNA's properties. So cofactors used in ribozymes include vitamin B12, FMN, glucosamine 6-phosphate, etc. Some of them are used by the protein enzymes for oxidation, reduction and carbon-carbon bond formation even today. So what we should understand, lot of cofactors which RNA enzymes made use of are also being made use of by the protein enzyme, and their role more or less will remain the same as well. So they were also had several RNA molecules that is capable of something like this, and the answer is yes. There are plenty of such RNA molecules had such cofactor utilization, and some of them can persist even today, and the answer is yes. A lot of them persist even today and they perform the task more or less similar fashion. Why do we then have protein catalyst? We need to have an explanation for that. We have seen group 1 introns active site is mechanistically equivalent to the DNA and RNA polymerases, which we see in protein enzyme. DNA and RNA polymerases are nothing but protein enzyme, and the group 1 introns active site resembles a lot to that of mechanistically equivalent to that of DNA and RNA polymerases. What selective pressure led to the current protein-based system for replication and transcription? Because in the modern world, no DNA replication happen without a protein, no RNA production happen without a protein. That means the job which was done by the ribozyme is now almost completely taken over by the protein enzyme. Is there a reason behind it? And the reasons might be much greater than what we can assume. One could be fidelity. Fidelity, how accurately a job can be done. And then comes the processivity, how accurately the processing of the job can be done. And then reaction rate, how effectively a RNA can be produced. And then functional repertoire, that means the interaction can be much divergent. That means RNA has only 4 bases, A, C, U and G, whereas proteins 20 amino acids are there. And each of them, when you say basic amino acid, not that having a constant basicity, it can have less basic, more basic, extremely high basic, like that. Or if you say acidic also, it is not just in a fixed manner. So this varies quite a lot, and this allows the fidelity, processivity, reaction rate and functional repertoire is possible. That could be one of the reasons why the RNA world slowly switched into protein world. Now let us quickly look into the future of the ribozymes. Can we adopt newer and newer function to the ribosome? The in vitro molecular evolution of RNA is very much possible with the help of ribozyme. And we can also cause high throughput screening. That means you can design ribozyme and you can do a screening. And based on which you can create ribozyme based therapies are very much possible. So let us quickly see some of the emerging clinical applications of ribozyme. And it's an alternative or a combination with siRNA. siRNA is small interfering RNA. We will see more in detail how they are targeting an RNA because we haven't discussed about it. But time being, you understand, siRNA can also negatively regulate gene expression. That means siRNA can inhibit an RNA's expressivity. We will see more in detail when we study siRNA. So time being, we are addressing only the ribozyme. So 4 main categories that can be made use of ribozyme in clinical application. 1 as gene inhibitors, 2 as gene amenders, 3 as protein inhibitors, 4 as immunostimulatory RNAs. So these are all the 4 categories in which ribozymes can be made use of. Gene inhibitors means disrupt the function of a gene. Gene amenders means you tweak it, like you don't want that much of expression of a gene, reduce it, like you are increasing or decreasing the volume of a phone or a audio device. And protein inhibitors, that means certain protein alone can be inhibited. You don't want to degrade it, but you can inhibit it. And then immunostimulatory RNA, and certain ribozymes can be used to induce immunogenicity in the host so that it can provide protection against certain foreign RNAs and proteins. So some of the clinical applications of trans-cleaving ribozyme, let us see. Ribozymes clinical trials were in progress, it's going on. One of the anti-cancer approaches, VEGF. We have also seen about the degradation of VEGF in the earlier class, and also anti-HIV or antiviral treatment against the HIV virus which causes the AIDS. VEGF, I told you that it can cause cancer. When there is too much of VEGF and its receptor is present inside the tumor, the vasculature can form very effectively and cancer survives, and you don't want that to happen. So what happens here is, if you have a pathogenic RNA and you design a ribozyme that is a trans-cleaving ribozyme, it will go and bind, as you can see here, at specific loci, and it causes a breakage. So this is a chemically produced hammerhead ribozyme. It can cause a cleavage onto the target RNA. Then another example, how do we produce, how you deliver it? So you have a patient and you collect the cells from the patient, that is CD4 or CD34 cells you collect from the bloodstream, and you introduce this ribozyme in a vector into these cells and deliver back into the same patient, so that in the bloodstream the person has got this ribozyme and it can cause a protective effect against VEGF or against HIV. So the results are the transduced cells, as you can see here, transduced cells into the CD4 and CD34 cells are well tolerated and persist in the patient. And second result is transduced cells may possess transient survival advantage over controlled cells. Both are good. And the shortcoming, the long-term efficacy is still a question. So every time you may have to keep doing this, but it's a life-saver approach. And the efficient gene delivery system also is a question. Can we deliver very effectively? So these are all the things to be addressed when you want to come into the effective therapeutic intervention using ribozyme. Another example is employing trans-splicing mediated repair of mutant transcript. So here what happens, you have a mutant transcript. That means, like many such examples are there. Duchenne muscular dystrophy, you know that mutation causes very severe case of organisms, muscles, and the person doesn't survive, and it's very much affected human males. So let us think such an example, a mutated transcript, and you want to repair it. So you can make use of a ribozyme, and that is having a corrected exon. The mutated part is now repaired and ribozyme has it. And this ribozyme can cause selective cleavage onto the place where there is a damage, and it can cause a splicing because many ribozymes have got a ligase activity also. So it cuts and then ligates. And this ligation happens with the fixed, correct, or the normal exon which is carried by the ribozyme. One way, many cancers are caused because of the mutation in the gene called p53. If you correct the p53 gene, the mutated p53, you may get a cure to the cancer because p53 is a guardian of the cell. And this way, this trans-splicing approach can help a lot in fixing in vivo. And like you can see here, if you have a exon and exon mutated one, you corrected it, fixed it here, and you end up getting a corrected RNA which can function quite effectively. So the concept is exploit the group 1 introns to cut and paste exons in a directed fashion. What we saw, how group 1 introns are functioning. The ribozyme is linked to a wild type gene fragment, and the corrupt means damaged, cis-splicing by inserting a wild type exon is very much possible. One such example is smart spliceosome mediated RNA trans-splicing. So this is a way in which you end up fixing damaged exons. So those who are interested can read about evolutionary insights into RNA trans-splicing in vertebrates and spliceosome mediated trans-splicing. These are all interesting articles to read. So in clinical trial, ribozymes and a lot of pharmaceuticals are there. Ribozyme and pharmaceutical incorporated, and angiozyme is a product which is used to target VEGF, and hepatozyme is a product that is targeting against hepatitis C virus infection. So these things are quite handy for the repair of the. And in clinical trial, anti-HIV therapy also is going on with bone marrow sample and to treat stem cell with retroviral vectors, and it encodes gene for anti-HIV ribozyme and reimplant the treated cells. So these are all the way in which you carry out ribozyme mediated treatment. To summarize, ribozyme challenges the central dogma of biology. Tertiary structures stabilized by conserved H bonding regions also possible, and divalent cations are required for the catalysis of the ribozyme. And ribozyme offer great pharmaceutical promise in general. And with this, I end the ribozyme part, and we will see a new topic in the coming classes. Thank you.